Today we will discuss about what are the receptors that suppress these T cells activations via ARFGEF1. We have understood that when this ARFGEF1 is not there, then the suppression of T cells reduces to a greater extent. We have discussed that thing in our previous videos. You can refer to those previous videos. In this video we will discuss the reasons or the receptors that plays a role towards these T cell suppressions via ARFGF1. Now the heading it's clears that it is TXA2 that might be or that might plays a role towards these T cell suppressions. So how they come up to this point? So from the previous literatures they are able to find out that it's the GPCR that plays a role towards these T cell suppressions, that plays a role towards this interactions with these T cells. So they trying to check the bioinformatics of these T cells in the presence and absence of in the naive and activated T cells, and check the interactions of this G alpha 12 and G alpha 12 and 13 coupled GPCRs. And out of this what they are able to see is that in the case of activated T cells, 18 of these G alpha 12 3 coupled receptors are found, and those 18s are shown here. And we can see that the level of expressions is not uniform for both naive and activated T cells. So they wanted to check their corresponding ligands and their analogs, and check the impact of this activated T cells, and in the case of wild type and ARFGF1 knockdown. And what they are able to see, most of the ligand does not show any significant changes except this TXA2. TXA2, they can see that the changes are statistically significant. That is, activated T cells goes down for the wild type, but for the ARHGEF1 it does not have any impact. And when they checked its impact with different concentrations of this analog, and this impact they have seen using this analog, that is U46619, which is the analog for TXA2. And when they check its impact at different concentration of this TXA2 analog, they are able to see that activated T cells goes down with increase in the concentration of this analog, and CD8 plus T cells signals will also goes down. And these things are shown. This the CD44 plus and CD62L plus ligand goes down with this TXA2 analog increase in the concentrations, and the CD44 and CD62L plus ligands goes up with this increase in the concentrations of this TXA2 analog, but there is no impact has been observed on this ARFGF1 knockdown, indicating that the signaling pathways is not completed in the case of this ARFGF1 knockdown, but it is completed in the case of this wild type T cells. And these things they have shown using the flow cytometer results are also shown, where we can see that in the ARFGF1 knockdown the changes are not statistically significant. It's around 94.2 to 92.5, and when they use this TXA2 analog. But when they use in the wild type, the CD44 plus cells is in the wild type without TXA2 analog is around 84.5, and it reduces to 44 when they use this TXA2 analog, indicating that it's the TXA2 analog that plays an important role towards CD8 plus activations. And then they check the CD8 plus T cells. This I have by mistakenly explained previously, but now is the right time to explain this panelly. In this case, the activated CD8 plus T cells, we can see that its concentration also goes down when they increase the TXA2 analog concentrations in the wild type T cells, but not in the case of ARFGF1 knockdown. And when they overexpress the receptors that plays a role towards this TXA2 ligand, that is TBXA2R and they can overexpress the activated CD8 plus T cells in the wild type, but there is no changes in the ARHGF1 knockdown cells. This all results indicates towards the point that TXA2 receptors plays a role or activate the signaling pathways that interact with this ARHGF1 to cause suppression of T cells. And when they check the photomicrograph of the cells, in the case of vehicles, we can see there is no changes. But when they use TXA2 analog in the wild type, we can see there is a change in the morphology, but there is no change in the ARHGF1 knockdown cells. And when they use the TP inhibitors to prevent the signaling pathways, even in the wild type, there is some recovery has been observed compared to the TXA2 analog, but there is no change in the ARHGF1 knockout. So this further substantiates their finding that the signaling pathways plays a role towards these receptors, that is TBX2AR, through these interactions of this ligand, that is TXA2, and thereby triggers signaling pathways that interact with this ARHGF1 to suppress the activations of the T cells. So I hope you understood how they validate how they come up to this point that the TXA2 that suppresses T cells activations via ARHGF1 through this TBX2R receptors. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.